This video is the presentation of Dr. Duaa as delivered in the International Symposium Food Product Development using less utilized crops, fruits and food waste, quality and safety, held on 2nd June 2025 at Sultan Qboos University, Muscat, Oman. She is Assistant Professor of Nutrition and Psychological Science, King Abdulaziz University, Saudi Arabia.
Assalamu alaikum wa rahmatullahi wa barakatuh. Today, inshallah, I will share some of the findings I found through my PhD in Newcastle University in the United Kingdom. I'll talk about the background, the hypothesis, the aim, and some of the empirical work I did during my PhD journey. Okay, so as we all know that dates have cultural and nutritional values in the GCC. We eat a lot of dates, and we like dates, and we have this beliefs that this food has and cognitive functions, but without any scientific evidence. Like other products or crops like blueberries, cocoa, raisins, they all have very established scientific evidence in the literature. Scientific evidence in the literature. And since I'm Saudi and we produce and we eat a lot of dates in Saudi Arabia, when I went to the UK to do my students who have their own proposal, and no one was interested in dates except me. That's why I had a lot of challenges and I suffered a little bit during my PhD. So, as you know, dates is, we have more than 3,000 different cultivars in Saudi Arabia. And I was very, very amazed by the work that has been done here in Sultan Qboos University regarding date, and I'm sure you will be very familiar with these names. In exploring the effect of dates on from a neuropsychological point of view, but however, still there is a lack in the literature regarding this field. Also, in terms of investigating the effect on animal, there are also some literature also has been done here in this prestigious university. Okay, so my objective were, since there was a lack of literature review regarding the dates and cognitive functions in human, my objectives were as following: to develop or create date-based food with a standardizable food phenolic content, to investigate it in human healthy volunteers, young healthy volunteers, and also to use the seeds, the roasted seeds, to make light coffee beverage, and also to investigate the acute effect on healthy young volunteers as well. But I faced so many challenges, like how should the treatment should be developed, what type of dates shall I pick. We have, as I mentioned, more than 3,000 different cultivars in Saudi Arabia. The same goes for the seeds as well, which seeds shall I pick. And these challenges were due to the novelty of the research question, inaccurate published data regarding the phenolic content, and also my limited ability to criticize the literature. One of the professors previously mentioned that we should not truly rely on what has been published in the literature, because sometimes there is a little bit of exaggeration or a little bit of, please don't quote me, sometimes they are a little bit misleading. And also, I wanted, I was a little bit biased. I wanted to find what they have found. What they have found in blueberries, raisins. I wanted to find the same thing in dates because I cannot lie. I was very biased. I'm still biased to dates. So, I followed a very robust methodology to do this research. I was thinking how to convert this material into standardizable, eatable form, and how to form and make the placebo to conduct an RCT. I screened several date cultivars, and the question was, what was the dosage? We eat a lot of dates, but it has been estimated that the consumption in one sitting per day was 115 grams per day in the Gulf countries. So I used this dose. Also, it was really difficult to mask all attributes in the placebo, because you know the date has a very unique taste. And to make a placebo that has the same taste, it was a little bit difficult. So after screening different types of dates, I used al-khsab and al-barhi, which are very famous in Saudi Arabia, and then I used the freeze drying machine to make it freeze dried powder, just to help in standardizing the treatment. Then I did some research to quantify the phenolic content, and then all the recipes were finalized, including the placebo, and I did sensory evaluation to make sure that the participants cannot differentiate between the three. The same thing goes for the date seeds. I screened 6 different varieties of date seeds. I did the phenolic quantification for the raw and the roasted date seeds just to find the difference, but I could not use the raw one to make the coffee. That's why I go with the roasted one. And surprisingly, the British people loved this coffee, loved it very, very much, and they kept asking me how can we get it or from where can we buy it. So it can be a commercial product. Although we have it in Saudi Arabia, United Arab Emirates, they sell it some shops, but we should invest more in this product. So after finalizing all of the treatments and the placebos, then test them for sensory evaluation and keep on modifying the recipes until we make sure that none of the treatment are highly differentiated from each other. I moved to the second part of the study where I tested the cognitive functions. So I conducted 2 studies, both followed double-blind control, powered by the most relevant studies. I registered them in clinical trial, and then I used a sensitive cognitive and mood tests, then used the proper statistical analysis, and I managed to test them in different time intervals according to the concentration max intervals, according to the concentration max of the phenolic content at that time. So the first study was about the date seeds. I used the yogurt to work as a media to mix and this is the content of the treatment. Most contents were matched except for the phenolic content. So the sugar, the fiber, all the ingredients were matched except for the phenolic content. A tool to assess cognitive function. It has a lot of tasks, but the outcomes of each task can be combined together to make cognitive index. As you can see, the choice reaction time, special sabatial working memory, and stuff like that. Special sabatial working memory and stuff like that. So the outcomes can be combined to make those 8 indexes. The same thing goes with the mood test. I used the Bone Lader mood scale. It's a very old and validated scale. And the outcomes of the 16 questions will be combined together to make 3 factors, which are alertness, calmness, and contentment. The study protocol, all participants should come at 9 o'clock in the morning to the research area in Newcastle University without breakfast to do the baseline, and then they will be given the treatment of the day. All treatments were randomized, and a third party was responsible for making the treatment distributed or given to the participants. Then we allowed them for 45 minutes for absorption and bioavailability. Then the same tests were repeated at 45 minutes, 90 minutes, and 135 minutes. The second trial has followed the same protocol, except that this time we increased the sample size to 49 healthy participants, and this time it was about the date seeds coffee. Again, the same thing. The placebo mainly was hot water and brown color. That's why the sensory evaluation test wasn't very, there was a significant difference between the 3 treatments, not like the date containing treatment, because of when I analyzed the coffee flavor that was available at the market at that time, it contained some caffeine and it contained some phenolic content. So we excluded the flavor and we stick to the hot water and brown color. It followed the same protocol, but as I noticed that from the previous study that keeping the participants until 135 minutes was a bit too long for them and they felt hungry. During the trial. So the results, as we can see here, we can see some significant results. However, I decided to go with a very cautious approach to analyze my data, since the larger of the outcomes was really big. So I decided to go with a cautious approach and to do some correction to the p-value to be very accurate in reporting my results. So after the adjustment of the p-value with Banfaroni correction, only the Bone Lader mood factor, the alertness, remains significant. The same thing has happened with the date seed coffee. There was only one significant result, and after the correction, none of the result remains significant. So I had to choose either to the result without the correction, as most studies in the literature, or to stick to my approach and to add a value or to make a difference and help new researchers like me not to invest time and money and just to follow the literature without criticizing without being very, very cautious in what to follow and what not to follow. So I decided to go with the p-value correction, because this is made me go back and read the literature with more knowledge and more. I become a little bit mature, so I was able to figure out why did they find something and I could not. So I found that most of these studies did not identify their primary outcomes, and therefore they did not correct the p-value. The reproducibility of the study wasn't really achievable, and also there was a problem with the tool sensitivity. Most of these studies did not register their trial and clinical trial registration, which also contribute to not identifying their primary outcomes. And I put some examples in these tables. As you can see here, for example, these studies found significant effect for caffeine. Since the second trial, there was a date seeds and a placebo. And I assumed that my robust methodology should shown the same result, at least for the caffeine, but this did not happen. So I went back and see that they did not correct for the p-value, and also there was no information about the registration. Also for the phenols studies, there were no corrections, and also most of the study did not publish their registration number. Also, the reproducibility of these studies were really a bit hard, because there was inconsistency in the result, dosages used, and also inconsistency in the tools. As we can see here, a huge dose like 1,470 showed no effect, while a small dose of 150 showed better performance and reaction time for attention task. Also 500 reaction time for attention task. Also 500 milligrams showed an effect, while 200 or 500 milligram showed no effect. As regarding the tools used, there were a variety of tools, although most of them test the same tasks or the outcomes. This is also contribute to the inconsistency seen. Also, I found that the tool sensitivity depends on the population. For example, the studies that tested elderly volunteers showed significant results. But when it comes to young or healthy volunteers, which is obviously easier to get the ethical approval, to process, to find the participants, there was a huge difference between them. So, as you can see, old volunteers, young volunteers. The difference here was in the type of the tasks. So when they tested young volunteers, they tend to use high-intensity tasks to create like stress or to reduce their baseline. Then they provide them with the treatment and then reassess, which mainly they're testing. They're testing not the effect of the treatment on the baseline. They lower the baseline, then they test it. Significant results I found, the utilization of robust methodologies and the absence of enhancing effect even of the positive control, which is the caffeine. Also, with the consideration of the inconsistency in phenolic dosage, primary outcomes, and sample age. Actually, as I said, we ate a lot of dates, and we believe in dates. And since we could not even prove the caffeine, that doesn't mean people will quit their morning cup of coffee. It's the same for us. We will keep on eating dates, and we will keep on make more robust research on dates, regardless of the insignificant result. And also, the insignificant results should be published and should be circulated, because they add to the knowledge and they add to the state of art. So this is the first study. This is the first study or project to develop standardized phenolic quantified treatments derived either from dates or date seeds to evaluate their effect on mood and cognitive function in young. This is the barcode where I have my ResearchGate, LinkedIn, to see more of my publication and connection. Thank you very much.
Thank you, Dr. Duaa, for the nice presentation and the experiment that you have gone through with the date and the date seed. We start collaborating with her, and inshallah we'll give her some date seed powder from Oman, from Mepsili, also to try. And she's having some idea where you can add in the, is it muffin or?
We have this a very traditional recipe of date cake or muffin. We want to add the date seeds powder to fortify it for fiber and phenolic content, needed to the military to see the effect on cognitive functions.